So this is all very interesting, but these observations and insights haven't been sufficient to sort of elaborate sort of a molecular pathway or signaling pathway in how mitochondria is sort of causing Parkinson's when they're damaged. Certainly, what it does highlight is that in Parkinson's, one of the major cell types affected are dopamine neurons, and these are highly energetic cells in the body. One dopamine neuron innervates thousands of neurons in the striatum, so this requires a lot of energy, suggesting that maybe these neurons are vulnerable to changes in mitochondrial function. But nevertheless, the mechanisms weren't really clearly elaborated from studying sporadic. And what has really aided the field has been understanding rarer causes of Parkinson's, the monogenic. And where I thought people might be interested in is that this is actually the 20th anniversary of this study from my former PhD supervisor, Nick Wood, where an Italian neurologist, Enza Valenti, had traveled to Sicily and found a family called the Marsala kindred, in which there were a number of consanguineous loops leading to autosomal recessive Parkinson's. And there was sufficient cases for Enza and Nick Wood to map this to chromosome 1p, which was a novel locus at the time. And as Dorothea mentioned, during my PhD, I was involved in the discovery of the actual gene in that family, and this was the PINK1 gene. And the family turned out to have a truncating mutation, this tryptophan 437 with a stop codon, and it was through the kinase domain. And after the initial report, most of the families that were then found to have PINK mutations had mutations that were located with the kinase domain. And so I became more interested in understanding the role of the catalytic properties of PINK1. And so that is what led me then to start my research lab and questions. And the question was at the very beginning, so PINK1 is a protein kinase. It was actually the first protein kinase linked to Parkinson's. And my question was, well, what are the substrates? And as Dorothy already mentioned, it took us a few years, but we were able to find very robust evidence that PINK1 can phosphorylate another Parkinson's protein, Parkin, and it does this at its N-terminal ubiquitin-like fold at this conserved serine residue. And we and other groups then subsequently found that PINK1 can also phosphorylate ubiquitin at the similarly conserved residue. And it turns out this is activated upon mitochondrial damage, and that the phosphorylation of these two proteins leads to the activation of Parkin. And this work was actually very useful in terms of understanding this breakthrough finding by the ULab, who had found that when mitochondria are damaged, the ULab found that Parkin was recruited to sites of mitochondrial damage, and that this led to the elimination of damaged mitochondria. So where I think this is led to is, I think, very good understanding of how cells now recognize and at least one pathway by which they can sort out and remove damaged mitochondria. And this is sort of outlined here, and this is just a sort of a figure, but these phosphorylation events that I've mentioned are critical for the recruitment of Parkin to the mitochondrial surface. And when it associates with phosphoubiquitin and becomes active, this leads to the ubiquitination of many substrates at the mitochondrial surface, and then the subsequent recruitment of the autophagosome machinery. And this leads to the elimination of the mitochondria through autophagy. So this has been extremely well studied, and reproduced in many different independent labs. But what we were very interested in was that much of this understanding had been gained in artificial systems, human cancer cell lines, for example, in vitro systems, very beautiful structures also produced validating this model. But we were very interested in knowing how relevant is this to the Parkinson's sort of pathogenesis. And so to address that, we decided to generate a knock-in mouse, in which we mutated serine 65 to an alanine. And these are just neurons from these mice that were stimulated with mitochondrial depolarizers. And you can see here that if we look at Parkin activity through ubiquitination of this substrate SysD1, we can see complete loss of activity. And this in the mouse was associated with region-specific mitochondrial deficits, particularly in the striatum, where we saw decreased respiration. But the mice itself is not a model that's been particularly useful for looking at the pathology. And so we benefited greatly from collaboration with Anu Vartuvara, who had discovered in Finland a patient who was homozygous for a mutation at the same site, serine 65 to an asparagine. It turns out this is a very rare event because the Broad exome database reports that this mutation has a carrier frequency of 1 in 60,000. So the chance of finding a homozygote is about 1 in 3 billion. But nevertheless, through Anu's patient and also through the PPMI initiative funded by the Fox Foundation, we were able to find 2 patients. And these patients were sequenced for other genes. And so, for example, in the Finnish patient, over 80 genes were sequenced, and the only mutation found was this at the phosphorylation site. And this patient subsequently had a brain scan. And this, normally what you see when you do these functional brain scans, you see a very nice crescent-shaped uptake in the brain. And now you can see here there's 2 dots suggesting degeneration. And the patient had a very, very classical phenotype for Parkin with early age of onset and sustained response to L-DOPA. So this really, I think, confirmed that the axis by which PINK1 activates Parkin is critical in the development of Parkinson's. And I think that's been quite a useful observation in terms of clinical and translational validation. But what this work made me wonder was, so obviously Parkinson's is a brain disorder, but what are the key substrates of Parkin in the CNS? Now, to address this, we teamed up with the Harper lab. So Wade Harper's lab has been at the forefront of proteomic technologies for identifying ubiquitinal modifications. And his lab had provided us with great knowledge on Parkin substrates in systems such as HeLa cells and other cell lines. And more latterly, his lab has been working on human eye neurons. So we decided to team up and use the Harper expertise to probe unbiasedly ubiquitination in a mouse neuron system. And so this was work that was led by Odetta Antico, a PhD student in my lab, and Albin, a postdoc in Wade's lab, who's now gone on to start his own lab at Sloan Kettering. So the system that we like using in Dundee is we like growing these cortical neurons from E16.5 embryos. And these, so this is MAP2, which is a neuronal marker, and this is GFAP, which is a glial marker. So these are very, very highly enriched neuronal cultures. And these actually are turned out to be a very good system for studying Parkin. So we took these neurons and with the help of Raju Nirojogi, who's a postdoc in my colleague Dario Alessi's lab, we did some copy number analysis, and in neurons in the system, there are about 18,000 copies of Parkin. This is also actually a very good system for studying other Parkinson's genes. There's actually many more copies, for example, of synuclein and VPS35. But we took these neurons and we wanted to analyze them for ubiquitination. So this is sort of the biochemical picture that you can get from these neurons. So these are wild type and pink knockout mice. And if you take neurons from the wild type mice, you can see if you stimulate and damage the mitochondria, you get very robust stabilization of PINK1 and phosphorylation of ubiquitin and also Parkin. And this is all lost in the knockout neurons. But one of the things that we observed early with Wade's lab was that, in contrast to what had been seen in other systems, if you look at the different linkages in these neurons, the one linkage that appears to be upregulated is mainly K63. the Harper lab has also found to be predominantly the case with endogenous Parkin in human eye neurons. So we next took these neurons and did unbiased diglide proteomics. And this is a volcano plot of wild type neurons, which have been stimulated with depolarizers for 5 hours. And we found amongst the top proteins that are being ubiquitinated, these are predominantly mitochondrial. And we can look at them in a bit more detail. So if we look at the types of mitochondrial proteins that we see, the majority of these are located at the outer mitochondrial membrane. We also repeated this proteomic experiment using wild type and pink knockout neurons. And what I'm showing here is a representation whereby on the y-axis, this is the knockout neurons, and on the x-axis, it's the wild type neurons. So anything that's to the sort of the right of this line are substrates that are enriched in the wild type neurons. And what you see here is that all of these mitochondrial proteins that are being ubiquitinated are occurring in the wild type neurons, and you're not seeing anything here in the knockout neurons, suggesting that these are indeed sort of pink dependent ubiquitination sites. So the next thing that we wanted to know was, so this was obviously exciting from a proteomic point of view, but can we also detect these sites biochemically? And the way that we did this was to set up this reconstitution assay of intact mitochondria from MEFs. And we've just deposited this protocol in Protocols IO if anybody's interested. But what we do is we take intact mitochondria from MEFs. These were wild type MEFs or pink knockout MEFs. And then these MEFs are either untreated or stimulated with depolarizing agents. And then we perform an in vitro reaction of these mitochondria with recombinant Parkin. So when you have no stimulation of these mitochondria in these MEFs, you get no activation of either pink or Parkin. But if you, what we noticed was that if you do stimulate the MEFs and look at the mitochondria in the absence of Parkin, you can see that there is phosphorylation of ubiquitin that's mainly monoubiquitin. But if you incubate this mitochondria with Parkin, you now see that this is now lost with now you see a high molecular weight smear indicative of the incorporation of this phosphoubiquitin in substrates at the mitochondria. And these events are all completely abolished in the knockout neurons. So with this system set up, could we now observe ubiquitination of these substrates detected by the mass spectrometry? And the answer was yes. So this is just a snapshot of some of the ones that we could robustly detect. So CysD1, CYBP5P, CPT1, you can see here only when you have recombinant wild type Parkin and activated, pink activated mitochondria do you see band shifts in these proteins indicative of ubiquitination. And these are completely lost in the knockouts. So this is a validation of our mass spectrometry data sets. We also, for a number of these substrates, could also look completely in a cell-based manner at ubiquitination using the endogenous Parkin in pink 1. And so this is a time course of neurons in which we do a tube pull down using this yeast multidisc protein to capture ubiquitinated proteins. And if you blot for either CysD1 or CPT, you see this time-dependent ubiquitination. And you can see here that these ubictinations are completely lost in the knockout neurons. So this was, again, further evidence for this being Parkin dependent. So in this work with Wade's lab, we were able to establish that when we compared these substrates with the human neuronal substrates, we found 24 mitochondrial proteins that are common. And so we think this is a common set of proteins that could be used as a proxy readout for damaged mitochondria. And that we would propose that these also happen much earlier than when mitophagy could be used as a readout. And we think this will be very useful for example, companies that are developing activators of PINK1. And there are also a number of companies interested in developing PET ligands as biomarker tools. And there's already one ligand, for example, against complex 1 subunit. So we think that these could also be attractive for this. So this work has provided some more detail on the proteins at the surface of mitochondria of neurons. But one of the things that we've also been interested in is what are the early steps by which PINK1 is activated by damage? And this is something that is, I think, now becoming of increasing interest as we now are sort of in some ways learning about the pathway. And just to give you some context to this work, many years ago, we and others had found that if you try and express human PINK1 in E. coli or in insect cells, this is just a kinase assay, there is no activity that you can detect. But if you, and this is one of the reasons why we switched to the orthologs, is that this is in contrast to insect orthologs, which are catalytically active. So we didn't really know why these pink proteins were inactive. But we did speculate that perhaps PINK1 has to be activated in cells by the damage, and what's the mechanism of that. So as Dorothea had mentioned, we had previously solved a crystal structure of one of these insect orthologs a few years ago. And one of the findings of that was that, that one of the sites of phosphorylation on the insect, serine 205, is required for substrate recognition and subsequent phosphorylation of ubiquitin. And so this serine 205 is very important, and it's also very conserved. So in the human PINK1, the serine 205 is conserved as serine 228. But in previous studies, we had done extensive phosphomapping mass spectrometry of human PINK1. We never observed phosphorylation of this site very easily, but we did find some other sites, such as serine 185, serine 187, but these were not regulatory sites. So Andrew Waddle, a postdoc in my lab, had noticed that the way that we extract PINK1 from cells with Triton resulted in still quite a lot of PINK1 left in the insoluble fraction. Whereas if you extract PINK1 with STS, Andrew saw that you can extract all of the PINK1. And so Andrew decides to do a phosphoproteomics experiment under STS lysis conditions. And this is just briefly the workflow, but what we did was we took HEK293 cells that express either wild type or kinase inactive PINK1. And under these conditions, you can see by phostag that there's phosphorylated PINK1. And so we process these samples for mass spectrometry. And under these conditions, we were able to confirm and see a peptide for phospho-228 and actually completely unambiguously assign that by as a serine 228. And this, I think, is the first time that that's been done. Previous papers have suggested this peptide is phosphorylated, but not specific for the serine 228 site. And you can see here in this sort of in this figure, this site was not found in the kinase inactive or in the unstimulated, suggesting that this is an autophosphorylation site similar to that of insect PINK1. So we've also, with in collaboration with the Fox Foundation and ABCAM, generated an antibody against this site, and this antibody is able to detect overexpressed phospho-228 PINK1 that you can see here in the cells with wild type, but it's not present in the kinase inactive and in the serine 228 alanine expressing cells. And so using this antibody, we've also been able to show that this site is also phosphorylated in endogenous PINK1. And so these are cells which express either wild type or CRISPR knock type for PINK. And you can see that we can see very clearly endogenous phospho-228. So this is providing strong evidence that in human PINK1, serine 228 is a critical regulatory site. And where we further demonstrate that is that we've studied the activity of human PINK1 with phospho-mutants. And you can see here that in these CRISPR knockout cells, if you replace wild-type PINK1, you get robust activation of PINK. But this is completely hampered by the serine 228 alanine mutation or an asparagine mutation. And interestingly, a nearby site on the insect PINK1 which is concerned in human serine 230. We don't think this site is phosphorylated in the human, and we don't see any impact on activation of PINK1. So one of the things that we've also exploited, knowing that 228 is phosphorylated, is use this Jason Chin technology from the LMB to express phosphoproteins in E. coli using a natural amino acid incorporation technology. And how we did this was we used a fusion construct of MBP PINK with a catalytic domain containing fragment of human PINK1, and we incorporated phosphoserine 228. And you can see here in the Kamasi stain of the phospho-228 protein that we had stoichiometrically phosphorylated protein. And what was very interesting for us to observe was that, as I previously mentioned to you, the wild-type human PINK1 in E. coli has very little activity. But now if you incorporate phospho-228, you can see that there's robust activity in vitro. And this, I think, is one of the first demonstrations that we can now observe human PINK1 activity in vitro. And we can also see that if you make mutations to block ubiquitin interaction, either with this point mutation G39D or removing the substrate binding loop, you block this effect. So these studies on phosphorylation now, I think, are indicating that serine 228 is a critical site for human PINK1, and we think that this is going to be a very robust biomarker of human PINK1 activity. And we think that the establishment of this in vitro assay for human PINK1 will be very useful for the field to study mutations which are not conserved in insect species. So there are a number of human PINK mutations that are not conserved in insect. So we think that this assay will be very useful for studying the effects of these directly. So one of the questions that we've had more recently is, how is it that PINK1 activates in a way to autophosphorylate 228? What are the sort of requirements of PINK1 for mediating that? And as I previously mentioned, the crystal structure has been very useful for us in studying and understanding substrate recognition. It was also very useful for us because it allowed us to understand how many disease mutations affect the kinase activity. But the insect structure that we solved began at residue 150. And so it didn't address more upstream elements than PINK1 in the N-terminus. But one of the recent projects that we've been doing is to try and understand what is the minimum domain requirements of PINK1 for activation in cells. And we've done this using a trick whereby we've substituted the N-terminus of PINK1 for a small MTS from HSP60. And we find that this can successfully target PINK1 to the mitochondria and become and allow it to become activated. And we've carried out extensive mutagenesis studies on this fusion protein. But what's very interesting is it's allowed us to basically pinpoint the minimum requirements. It turns out that you need PINK1 to start from about residue 111. If you start at 112, 13, it's completely dead, although the proteins here are expressed and go to the mitochondria. So what is it that's potentially between residue 111 and 150 that's so important for PINK1? So in previous reviews of PINK1, this region has been sort of very loosely termed as a sort of a linker region, but it does have some conserved elements. And what's interesting about this region as well is that there are a number of disease mutations. So there's one at cysteine 125, and there's also one at glutamine 126 within this region. If you take the sequence of this region and just put it through sort of some prediction, secondary structure prediction sequences, it does strongly predict that there is an alpha helical domain within this. but we weren't able to obtain any experimental evidence initially from this. But what we've been very benefited from greatly has been the recent breakthrough in artificial intelligence from this DeepMind company, which have, in collaboration with EMBL, produced a very easy, friendly to use database called the AlphaFold, whereby you can search for it for nearly all human proteins, and the DeepMind AlphaFold program has been predicting the tertiary structures of these. And when you do that for PINK1, what you see here, so this is the structure here that's predicted. This is the same data, but we've just included the confidence assignment by DeepFold on the AlphaFold. And you can see here that this region where it started 111, which we're calling now the N-terminal extension, is predicted to be alpha helical, and this is with very strong confidence. And what was also interesting from this model was that this N-terminal extension was shown to be very closely associated with the C-terminal extension. And so we were able to do some experimental validation of this. So we took some insect PINK1, which contained this N-terminal region, and in collaboration with colleagues in Leeds, performed some hydrogen-2 exchange mass spectrometry. We strongly predicted that this was likely to be very well ordered, consistent with alpha helical arrangements. And then what we also have done is do some mutational analysis. So as I mentioned, there are some disease mutations here in the N-terminal extension. And what we've done is we've taken HeLa cells which have been knocked out for PINK1 and replaced them with point mutants in this N-terminal extension, and also a number of point mutations that have been reported in the literature in the C-terminal extension. So one was a leucine 539 to phenylalanine, and there was also another insertion of a Q at 534 to 535. And what you can see under our assay conditions is that these mutations in the NTE and also this 534 to 535Q completely abolish PINK activation. And they seem to be doing that through decreased stabilization. So if you look at the levels of PINK1 in these mutants here, they're significantly less. In fact, it's just under 50% than the wild type. And this is also associated with complete loss of the 228 phosphorylation. And this is contrasting with other mutations in the kinase domain. So for example, the G39D, which I mentioned, which is substrate defective. You can see here that there's no activation by ubiquitin phosphorylation, but you can see that there's quite normal autophosphorylation. And we see this also with another mutation, the glycine 49V, which is also predicted to interact with substrate. So this, I think, provided the fact that these mutations behave very similarly suggested that these could be linked. We've also confirmed this endogenously in collaboration with Julia Fitzgerald, and we've obtained fibroblasts from Tübingen, which carry the homozygous mutation, the Q126 to proline. This was from a family of 2 sisters reported in 2009. And you can see here that if you take these fibroblasts and stimulate them with mitochondrial damage, we completely lose all phosphorylation of ubiquitin, and that's associated with decreased stabilization of PINK1. So what might the mechanism be? So previously, the Eurolab and the Matsuda labs had shown that PINK1 stabilizes at the TOM complex. I mean, we're learning a lot about the TOM complex now through amazing cryo-EM structures of this multi-subunit complex. And this is one structure taken from yeast from Yu Ying Park's lab. And when you perform Blue Native page analysis of PINK1, you normally see that the wild type stimulated PINK1 can be seen in a sort of 700 kilodalton complex. And we don't see this happening with mutations in the NTE. So we think that perhaps this is somehow impacting on stabilization of PINK1 at the TOM. So this draws me to a close. And so our current model of how PINK1 is activated is that we think that this alpha helical region within the N-terminus and the C-terminus collaborate with one another to allow PINK1 stabilization at the TOM, which then facilitates autophosphorylation of 228, and that this conformation is then able to allow PINK1 to phosphorylate ubiquitin and initiate the mitophagic cascade. And obviously this is a model and further work will be needed. But what's interesting is that the Trompe group at McGill have experimental evidence from tribolium PINK1 that this conformation predicted by AlphaFold is indeed seen experimentally in that insect. So with that, I'm just going to finish by I want to just maybe flag in the last section of my talk, Wally and Hina had performed several of the experiments that I showed. I thank all the members of my lab. This was a picture that we had recently, sort of as we start to ease out of COVID. I want to thank all my collaborators in these projects. We benefit greatly in Dundee from core facilities, and I'm grateful to all those teams. And I'd also like to thank all my funders, and I thank you all for your attention.
Thank you, Miratul. That was just a fascinating talk. I think there's a lot of questions, perhaps from the audience. So people who still have questions, feel free to send them in. But to start off, I'll take the opportunity to ask you one of the questions I had during your talk. So the one thing I was wondering is, when you made this HSP60 fusion protein with PINK1, you see that PINK1 retains its activity and has this ladder pattern of phosphoubiquitin. I was wondering whether you also specifically looked at the substrates that you identified with proteomics, whether you could hit sort of whether PINK1 was able to still reach the same substrates.
That's a great question. No, we haven't extensively done that. What we have done is we've found that other leader sequences of other mitochondrial proteins, such as ornithine, transcarbamylase, OTC, these can also substitute for the N-terminus in a way that it can allow PINK1 to be imported into the mitochondria, and also allow it to somehow, when depolarized, be arrested in sufficient conformation to allow it to be able to phosphorylate ubiquitin. But that's a great question, because there is obviously a hypothesis that some of the substrates that Parkin targets is somehow dependent on where PINK1 is physically in association with those subproteins.
And then maybe a follow-up question on that. Did you have any link in your proteomics data that you did with Wade Harper, whether the targets that you identified there, whether they are physically in vicinity of the PINK1 Parkin, similar to what Jean-François Trempe published just recently?
Yeah, I was aware of that. So we didn't extensively, in fact, we didn't do any detailed work looking at that. I mean, I think it's still a great mystery why, we see, in different systems, you see many protein substrates of Parkin. So it's obviously a very promiscuous E3, and certainly in vitro, it's very promiscuous. But nevertheless, that cell activity within the cell, within the intact mitochondria, is fascinating. I mean, I could give you lots of speculative answers. Abundance of protein, proximity, vicinity to PINK1 TOM complex. I think this is where maybe reconstitution would be useful as well in the future to try and drill down on that.
Then there's a question from Emma Gorst. She's a member of the Hurley Lab. She's asking, so first of all, she's saying wonderful work. She's asking, have you found any PINK1 specificity for different ubiquitin subunits in the chain? And related to that, whether there's a bias to specific ubiquitin chain lengths in primary neurons?
So in terms of linkages, we've previously found that, at least in vitro, PINK1 can phosphorylate all linkage types in vitro, at least dimers with all the linkages without any sort of preference. But certainly, Wade's proteomic and Albin's proteome analysis would suggest that, the key linkage appears to be K63, and so it must be, if one could look at that more specifically, and it's not possible at the moment, as far as I suspect that if one could analyze that, one would probably see K63 linked ubiquitin being preferentially phosphorylated, I would imagine. I mean, that would obviously be quite interesting to really confirm.
Yeah. Then I see there's 2 more questions. One's from Sylvie Urbe. I think we might be able to unmute her, Doro. Is that possible? Yes, I think Sylvie, you should be able to speak.
Yes, I just saw I can. A great talk, Miratul. I really enjoyed that. I was just thinking about the region of PINK1 that, you know, the TOM7 PINK interaction is important for, so that regulates import or rest at the TOM complex. Is that the same region that we're talking about here? The same helix?
Oh, so we've not experimentally done anything specific, but I think that in terms of where PINK1 could be associated with the TOM, I think the evidence from Richard Yule's lab that TOM7 knockout cells prevent PINK activation suggests that TOM7 could be the key subunit that is anchoring PINK1 when it's in the active conformation. And so I think it's going to be interesting for future studies to look at that, the TOM7 interaction with this NTECT helical domains.
Yeah, that's exactly what I wanted. Thanks. That's great.
Then I see that we've received a message from Wade Harper, sort of adding on to the question from before. He's adding that based on the clipase experiments, that most phosphorylation events are likely on the monoubiquitin or at the distal end of the chain. So perhaps there might not be chain dependence. I think there is one more question from Jim, and then I see there's one more in the chat for after Jim. Do you like to go ahead, Jim?
Right. I saw the question that came up in the Q&A, or one of them is actually similar to mine, so people are thinking along the same lines. So it's interesting to think about this model for mid-zone versus peripheral scission regulated by damage. I think that will actually be presented in one of the upcoming seminars by Suliana Manley, and I noticed, I think other people did also mitofusin in your list of substrates. So if you looked at whether there's any regulation of the balance between peripheral versus mid-zone fission.
We haven't done that, Jim, yet. No, we've not looked at that aspect of it. I mean, I think mitofusin, yeah, it's a very interesting substrate. Yeah, I guess for me, one of the things about the mass spectrometry data and also our reconstitution studies is that some of these ubiquitination modifications are occurring at very low stoichiometries. So whether these are sufficient to affect biological processes in a way that you can measure easily. But obviously, if you have very sensitive assays for that, then you might be able to see effects of mitofusin ubiquitination or not.
Okay, thank you. I think we've gone through most of the questions we have. That leaves me with, first of all, thanking you, Miratul, for a fantastic talk. And I would like to invite everyone for our next webinar series. I think Doro is still urgently trying to put up a slide deck for the next talk. So the next talk will be from Sasha. It's going to be on the 12th or 13th of October, depending on where you are in the world. If you haven't registered yet, please still do, but otherwise you'll get an automatic invitation for this talk as well. Once again, thank you, and have a nice day, everyone.
Thank you.
Thank you, everyone. Thank you, Miratul.
Thank you. Thank you again.